Hello everyone. In this session I am going to discuss about glycolysis. Glycolysis is an extra mitochondrial universal catabolic process by which glucose is converted to pyruvate under aerobic conditions in all living cells. During glycolysis, free energy is sequestered in the form of high energy molecule ATP. Glycolysis was described by Amden, Mayer and Parnas. Hence, it is also called as Amden-Mayer pathway. Glycolysis consists of 10 separate reactions, each catalyzed by a different enzyme. Glycolysis is regulated by 3 different control enzymes. The first of this enzyme is hexokinase, which phosphorylates glucose, turning it into glucose 6-phosphate. The phosphorylated form of glucose is incapable of leaving the cell through glucose transporter proteins in the cell membrane. The second is phosphofructokinase. This enzyme allows the production of fructose 1,6-bisphosphate. The final control enzyme is pyruvate kinase, which controls the rate of production of pyruvate, which is the final product of glycolysis. For each molecule of glucose that goes through the process of glycolysis, there is a net gain of ATP molecule, NADH molecule and water molecule. The pyruvate converted from glucose in glycolysis then enter into the citric acid cycle, which takes place in the mitochondria. There are 2 phases in glycolysis, which are preparatory phase and payoff phase. Preparatory phase. This phase is also called glucose activation phase. In the preparatory phase of glycolysis, 2 molecules of ATP are invested and the hexose chain is cleaved into 2 triose phosphates. During phosphorylation, glucose will be converted to glyceraldehyde 3-phosphate. The steps first, second, third, fourth and fifth together are called as the preparatory phase. Next one is payoff phase. This phase is also called as energy extraction phase. During this phase, conversion of glyceraldehyde 3-phosphate to pyruvate and the couple formation of ATP takes place. Because glucose is split to yield 2 molecules of glyceraldehyde 3-phosphate, each step in the payoff phase occurs twice per molecule of glucose. The steps after 5 constitute payoff phase. Reaction 1. The first reaction of glycolysis is catalyzed by the enzyme hexokinase, which mediates the transfer of ATP's gamma phosphoryl group to the number 6 carbon of glucose. The reaction occurs through the nucleophilic attack of glucose 6 hydroxyl group on the gamma phosphorus atom, yielding glucose 6-phosphate and ADP. This is the first of 2 reactions in which the free energy of ATP hydrolysis is invested in the pathway. In reaction 2, the second reaction of glycolysis involves the isomerization of glucose 6-phosphate and aldose to fructose 6-phosphate, a ketose. The reaction is catalyzed by phosphohexoisomerase. Reaction 3. In the third reaction of glycolysis, a phosphoryl group is transferred from ATP to fructose 6-phosphate by the enzyme phosphofructokinase. This brings the net investment for the pathway to 2 ATPs. The product of the reaction is fructose 1,6-bisphosphate. Reaction 4. In reaction 4, the enzyme aldolase cleaves the 6-carbon monosaccharide fructose 1,6-bisphosphate into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The glyceraldehyde 3-phosphate is released by the enzyme and proceeds along the glycolytic pathway. Reaction 5. The fifth reaction of the glycolysis pathway, catalyzed by triose phosphate isomerase, is the isomerization of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Reaction 6. At this stage of glycolysis, the original glucose molecule has been converted to two glyceraldehyde 3-phosphate molecules. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes the oxidative addition of a phosphate group to glyceraldehyde 3-phosphate to form 1,3-bisphosphoglycerate. This intermediate is then oxidized by the direct transfer of a hydride ion to NAD plus to yield NADH. Reaction 7. Reaction 7 is the first reaction of glycolysis in which free energy is recovered. In this reaction, the synthesis of ATP is driven by the hydrolysis of 1,3-bisphosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase and occurs by the nucleophilic attack of ADP's terminal phosphoryl group on the first phosphoryl group of 1,3-bisphosphoglycerate to yield 3-phosphoglycerate and ATP. Since two molecules of ATP are generated for each glucose molecule that entered the pathway, this brings the net ATP yield of pathway at this stage to 0. Reaction 8. In reaction 8, the enzyme phosphoglycerate mutase catalyzes the transfer of a phosphoryl group from 3-phosphoglycerate's third position to its second position, yielding 2-phosphoglycerate. In reaction 9, the enzyme enolase catalyzes a dehydration that converts 2-phosphoglycerate to phosphoenol pyruvate. In reaction 10, the final reaction of glycolysis is the pathway's second reaction in which free energy is recovered in the form of ATP. Here, the synthesis of ATP is driven by the high negative free energy of hydrolysis of phosphoenol pyruvate. In the first step of this reaction, a phosphoryl group is transferred from phosphoenol pyruvate to ADP to form enol pyruvate and ATP. The enol pyruvate intermediate then tautomerizes to form the reaction product pyruvate. This highly exergonic tautomerization provides free energy to drive the formation of ATP. Again, as two of these reactions occur for every glucose molecule that enters glycolysis, the yield of ATP for this reaction is 2 ATPs for glucose. Therefore, the overall yield of ATP produced in the glycolytic pathway is 2 ATPs for glucose. Net energy yield per molecule of glucose in glycolysis. Hexokinase, there is no ATP production. Phosphofructokinase, there is no ATP productions. In glyceraldehyde 3-phosphate dehydrogenase, you will produce one NADH2, okay, which is corresponding to 3 ATP molecule. Here you will get 6 ATP molecule. Phosphoglycerate kinase is having the capability of producing 1 ATP, and pyruvate kinase, then total net yield of ATPs is 8 ATPs. Energy yield in aerobic glycolysis. The conclusion: glycolysis pathway is essential for the following reasons. Glycolysis is the only pathway that is taken place in all the cells of the body. Glycolysis is the only source of energy in erythrocytes. The glycolytic pathway may be considered as the preliminary step before complete oxidation. The glycolytic pathway provides carbon skeletons for synthesis of non-essential amino acids as well as glycerol part of fat. Most of the reactions of glycolytic pathway are reversible, which are also used for gluconeogenesis. With this, I would like to conclude this session. Have a great time.